My name is Yuritsa Neri. I am a biology major at Ridley College.
And my name is Peyton Mayer, and I'm an electrical engineering student here at Ridley College. And together, Yuritsa and I are part of the Ridley College STEM Ambassador
Today
Program.
we're going to teach you how to do a DNA extraction. So first of all, you're wondering, what is DNA? DNA is the genetic material of all living things. So it's not just us humans, it's also animals, plants, bacteria, etc. This genetic material is passed from one generation to the other. So you have your parents' DNA, they have their parents' DNA, and so forth. For this experiment, we're going to extract DNA from wheat germ. So we know wheat is a plant, and we'll be able to see the DNA from that plant.
Now, before we hop into the video, DNA has a lot of functions. One primary function that DNA has is that it is able to locate ancestors of yours from past and prior generations. Another way that DNA is used is that it helps investigators find evidence of someone who may or may not have committed a crime at a crime scene. Now, onto the experiment. For this experiment, you're going to need a gram of wheat germ or a half a teaspoon of wheat germ, 20 milliliters of hot water or 4 teaspoons of hot water, a quarter teaspoon of dishwashing soap or hand washing soap will do as well, 10 milliliters of rubbing alcohol or 2 teaspoons of rubbing alcohol, a clear vial or a clear glass cup. You're also going to want a graduating cylinder, and if you don't have one of those, teaspoons will work, and a wooden stick.
So now we're going to get our gram of wheat germ and we're going to put it inside our vial. The wheat germ has cells and we're going to get our DNA from those cells. Then we will get our 20 milliliters of hot water and pour it inside the vial. The water allows the cells to flow around. And once we have done this, we will shake vigorously for 3 minutes.
After shaking for about 3 minutes, you are now going to want to take your quarter teaspoon of dishwashing soap, and you are going to want to slowly pour it down the side of your vial or your clear glass cup. This will allow your DNA cells from your wheat germ to be broken up, and it adds a density so it can separate layers. You are going to want to shake softly for about 2 minutes so you can avoid having as much bubbles as possible.
So now that we're done shaking, we see that there are some bubbles in our vial, and we will try to remove as many as we can with our wooden stick. So now we will pour slowly our 10 milliliters of rubbing alcohol so the alcohol can float and create a top layer. DNA doesn't dissolve in alcohol, so this will allow us to see it form as a white cloud in between the layer of the alcohol and the soap.
Congratulations, you have finished the process. At the wooden spoon level, you will see a white filmy layer, and that is your DNA.
And now, because we could never see DNA with our bare eyes, we put a sample of our experiment in an electron microscope. And here we are able to see our strand of DNA that looks like a worm. And now if we get a little bit closer, we can see it with more detail. And here's our wheat germ DNA.